Good morning. Welcome back to another video of Before Botany. In this video, we are going to discuss about cell cycle control system. We know the cell cycle, that is mitosis, and meiosis, along with gap 1, syn S and gap 2 phases. How these divisions are controlled in our body. These are discussed in this video. The progression of cells from one step to other in cell cycle is regulated by a series of protein complexes. So, the cell cycle is regulated by a series of protein complexes. This protein complex has two subunits, cyclin and CDK. CDK means cyclin dependent protein kinase. The cyclins are the regulatory component and the CDKs act as a catalyst, which is a protein kinase. In every eukaryote, there are different classes of cyclins defined by the stage of cell cycle at which they bind CDKs and function. For controlling the cell cycle, the cyclin must bind with the CDK, and at which stage these two binds together. Based on that, the cyclins are categorized into different types. Mainly, there are four major groups of cyclins: G1 cyclin, G1 bar S cyclin, S cyclin and M cyclin. Look into the picture where the red colored cyclin, that is the G1 cyclin, is where its concentration is higher in G1 phase. So it means that cyclin, G1 cyclin, is predominant in G1 phase. That's why it is called a G1 cyclin. And in vertebrates, the G1 cyclin is commonly called as cyclin D. But at the middle of the G1 phase, another cyclin compound is developed and it attains its maximum concentration in between the transition phase of G1 and S. That is the G1 S cyclin, or it is called cyclin E. Then during the S phase, the cyclin A or the S cyclin attain its maximum concentration, whereas in M cycle, that is at the mitosis stage, the cyclin B or the M cyclin is at higher concentration. So during the entire length of the cell cycle, all the cyclins are present in the cell itself, but during some stages its concentration are higher than the others, and at that stage they are acting, or they are the regulatory components. So there are four types of cyclins: G1 cyclin or cyclin D, G1S cyclin or cyclin E, S cyclin or cyclin A, M cyclin otherwise called as cyclin B. These cyclins bind to CDK and control their ability to phosphorylate their target protein. The cyclins are rapidly synthesized during a specific phase, and after they function, they break down immediately. So, during the stage in which the cyclin is needed, at that stage they produce at maximum rate. After its functioning, the breakdown happens immediately. Going to the CDK details, that is a cyclin dependent kinase, protein kinase. CDK also constitute a family of functionally related protein kinase. We know kinases are the enzyme that add phosphate group to the target. So kinases are a group of enzymes which add a phosphate group. And the target protein for CDK phosphorylation are determined by the association of cyclin. When the cyclin is attached to CDK, then only that complex is able to phosphorylate the target protein. Cell cycle has different phases, and each phases is characterized by phosphorylation of different target protein. And we know the phosphorylation events are reversible. When the Cyclin-CDK complex disappears, the phosphorylated substrate is dephosphorylated by phosphatase, another enzyme. So the phosphorylation is needed for the activation of that protein, and after the Cyclin-CDK complex disappears, that means we don't need that protein after that time. So we have to dephosphorylate that particular protein with the help of the enzyme phosphatase. And there are different Cyclin-CDK complexes called as G1-CDK, G1-S-CDK, S-CDK and M-CDK, that is G1 cyclin combined with a CDK, that is the G1-CDK. And in vertebrates, I said earlier, in vertebrates, the G1-CDK is called cyclin D, G1 cyclin is called cyclin D, G1S cyclin is called cyclin E, S cyclin is called cyclin A, and M cyclin is called cyclin B. In vertebrates, the CDK combines with the different cyclins, that is the cyclin D combines with the CDK4 as well as CDK6, and cyclin E combines with the CDK2, cyclin A combines with the CDK2, and cyclin B combines with the CDK1. And this is the diagrammatic representation where the Cyclin-CDK complex starts its action and the extent of its action. So the cyclin D, which will combine with the CDK4 and CDK6, is activated during the G1 phase. And the cyclin E, which is combined with the CDK2 and form the complex, which is start its action at the mid of the G1 phase and ends at the mid of the S phase. And the cyclin A combines with the CDK2 at its action at S phase, and it is prolonged for the G2 phase. And the CDK1 along with the cyclin A combines and form the complex, and it is mainly activated during the M phase. So these are the Cyclin-CDK complexes. Then how the Cyclin-CDK complex is worked? The cyclin undergoes cycles of synthesis and degradation. It is during the cell cycle, the synthesis and degradation of cyclin is undergoes by, whereas the CDK level remains constant. Whenever the Cyclin-CDK complex perform its action, we have to deactivate by the proteolysis of cyclin. So the Cyclin-CDK complex is deactivated with the help of proteolysis of cyclin. And the proteolysis is a ubiquitin dependent proteolysis catalyzed by an enzyme called a ubiquitin ligase. The enzyme S-CF, which is an E3 ubiquitin ligase, this is used for the destruction of G1 phase cyclin or the G1 cyclin. So ubiquitin S-CF, which is a E3 ubiquitin ligase. That is, ubiquitin ligase is responsible for the proteolysis of cyclin, where G1 cyclin is proteolyzed by S-CF. APC-C, that is the anaphase promoting complex or cytosol. This is the another ubiquitin ligase which is used for S phase and M phase cyclin, S cyclin and M phase cyclin degradation. And the degradation of cyclin is the degradation of Cyclin-CDK complex, or the deactivation of Cyclin-CDK complex can be done by the phosphorylation or dephosphorylation reaction, and binding of CDK inhibitor proteins, generally called CDKI. So this is about the cyclin and CDK complexes. Then we know during S phase, that is at the cell cycle, we have the S phase, that is a synthetic phase. During S phase, the chromosome replication occurs, that is the content of chromosome is doubled. How the duplication is controlled in S phase? Let's check. The S-CDK, that is the S phase CDK complex activation leads to the formation of a multiprotein called pre-initiation complex. So once the S-cyclin combines with the CDK, it becomes active, and that activation leads to the formation of a protein complex called a pre-initiation complex. And this pre-initiation complex unwind the DNA and begin the process of replication. And for this, the key player is the ORC, that is the origin of replication complex. And this ORC bind to the origin of replication. Then another protein called MCM protein, that is the mini chromosome maintenance protein, which is an hexamer like that of our helicase. MCM, that is MCM2, MCM3, 4, 5, 6 and 7. So it is an MCM protein is an hexamer which forms a ring around the origin. So we have that origin of replication. Upon that origin of replication, ORC bind on that origin of replication, and then the MCM protein also forms a ring around that origin. So another complex is formed called pre-replicative complex. After the formation of the pre-replicative complex, the origin is licensed for replication. So for the duplication of DNA in S phase, the MCM, that is the mini chromosome maintenance protein, acts as the licensing factor. This is just a diagrammatic representation. This is the origin of origin where the MCM, that is when the CDC6 and CDT1 binds with the ORC complexes, then only the MCM can attach to with that complex and form the pre-replicative complex. Then the role of M-CDK complex, that is the mitotic CDK complex. The M-CDK are commonly called MPF, that is the mitosis promoting factor or maturation promoting factor. And the M-CDK are synthesized during S and the G2 phase. The M-CDK induce chromosome condensation, breakdown of nuclear envelope, assembly of mitotic spindle and alignment of chromosome in metaphase. So these are the major events happens in the cell division. So for this processes, M-CDK is necessary. And for these processes, the M-CDK phosphorylate a wide range of proteins like condensin for the condensation, laminin which forms a stabilizing network under nuclear membrane, and sequerin which hold separase. So through these mechanisms, the cell cycle is controlled in our body, not in our body, in plant body as well as in animal body. So, thank you for watching.